In this video, we will be talking about blood types, mainly two types, the ABO group and Rh. These blood types are important for blood transfusion because if we give patients mismatched blood, that can be fatal, right? So it leads to, if they are mismatched, if the blood is mismatched, it leads to agglutination, which is the red blood cells, they come together and form big clumps, and that it is fatal. So ABO groups was discovered by Karl Landsteiner in 1930, and he got a Nobel Prize for that in 1930. So he gave a boon to transfusion, really, because ABO group is the most matched blood typing that we see, and also Rh, right? So that was discovered a little later than ABO groups. So blood types are explained based on antigens and antibodies. Antigens, in this case, the ABO groups, they are found on the red blood cells. So these are proteins found on red blood cells. And they're also born with antibodies which can react with the antigens. These antibodies are proteins found in plasma, right? So we have antigens on the red blood cells and antibodies in the plasma. So antigens are proteins, glycoproteins, glycolipids, which are very unique to an individual. They're found on the surface of all the cells. And when the body encounters these antigens, then they launch an attack by producing antibodies. Okay, so that's the normal process of immune system. And when the antibodies bind with antigens, then they form a complex called as agglutination. That is, they clump together and they clump the cells, whatever cell it might be. So when we talk about the ABO group, we are talking about presence of protein A, protein B, both the proteins or no proteins. Okay, so these are called the antigens, which are found on red blood cells, the surface of red blood cells. So A and B are glycoproteins found on the surface of RBC, and antibodies for AB are found in the plasma, right? So definite, and we are born with it. So of course, if a person is A type blood, has A type antigen, they will not have antibody A, right? So they will be born with antibody B, okay? So that it doesn't clump. And likewise, if they are given blood from another A, it really doesn't matter because they do not have anti-A in their plasma. If their blood is donated to somebody who has B type of blood, the B individuals will have anti-A, and that will agglutinate the A that this person is donating. So that's what we're going to mainly talk about. So let's look into, let me go back. Let's look into the four types and what kind of antigens they have and what kind of antibodies they have. A type will have A antigen and anti-B. B type will have B antigen and anti-A. AB will have A and B antigens, and that's why they do not have any antibodies. And O, who do not have antigens, will have both anti-A and anti-B. So if we go here, so we can see that type O has neither A nor B. Type A has A antigen, type B has B antigen, type AB has both A and B. What we write here are the genes, the alleles. Okay, so suppose you have taken biology, you must have seen this in genetics, in your genetics class, where you have two alleles, like these are homozygous alleles, this is heterozygous. Homozygous, heterozygous, we have only one type, A and B, and then this one is a recessive, right? So it doesn't have any protein. So as you see that it is genetically linked, right? So you have the gene or you don't have the gene, right, for A and B. In the plasma, you have antibodies. So O will have both anti-A and anti-B. A will have anti-B. B will have anti-A. AB will have neither. So if this person has to receive blood, they can receive blood only from O because they have both anti-A and anti-B. right? So if they receive blood from A, it's going to react with anti-A. If they get from type B, that's going to react with anti-B. If they get from AB, then both anti-A and anti-B is going to clump them. Let's take A. A needs blood, right? So they are getting the red blood cells. So they're getting the antigens. So as long as they get A antigen, they're good. So type A can get blood from O because O has no antigens. Type A can get from A because A is similar. Cannot get from B because they have anti-B. Cannot get from AB because they have anti-B. Same with type B. Type B can receive blood from O because O has no antigens. Type B can receive blood from B. Type B cannot get from A because they have anti-A. Type B cannot get from AB because they have anti-A, which is going to clump with this. When we look at AB, AB has no antibodies, so they can receive from anybody, right? Because they can get O, A, B, AB. They have no antibodies, so they're not going to clump any of these. So these are called as universal recipient. If you take O, if somebody wants to donate blood, then they can donate to anybody because they do not have any antigen. So these are called as universal donors. Type A, remember that they have A, okay? So they can give to A or AB because AB does not have any antibodies. They cannot give to O because O has anti-A. Type B can give to B or AB, cannot give to A because they have anti-B, cannot give to O because they have anti-B. AB has both A and B antigens, so cannot give to anybody except themselves. All right. So that is your ABO blood groups. And I'm going to add an animation from McGraw-Hill, and let me see if you can see this. Okay, let me play this.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
Okay, so I hope this makes it a little clearer. I like this one. I like this animation. All right. So this shows, sorry, I think I have, yeah. So this is blood typing. So if you do blood typing in the lab, you take anti-A, anti-B, and anti-Rh. So you take blood, a drop of blood, okay, put it on Petri dish, and then add anti-A and anti-B. So whatever coagulates, that is your blood type. So type A because it has clumped, type B because this is clumped, and this is type A and B, and this is O. Now let's talk about Rh group. Okay, so this is another table I really like. So you can see that this is type A, type B, both AB, and then this is O, anti-A, so right, anti-B, I'm sorry. This will coagulate with this, anti-A coagulates with this, AB have no, and O have both anti-A and anti-B. So A antigen, B antigen, AB antigens, and none, and then they're compatible with, right? So they can give blood to A and O. Sorry, they can get blood from A and O. This can get blood from B and O, and this one can get from anybody, and this one is universal donor. Okay, so this is a quick table that I really like. Okay, so I did write down all this, so you can watch that. And again, this is another picture that I really liked. So you can actually go into this and do the blood typing. And you can see that, sorry, excuse me. So this is a nice little table again. Now let's talk about Rh. So Rh group was found in 1940, a little later than ABO blood groups were formed. And Rh is positive if the person has the Rh protein on their red blood cells. This has nothing to do with ABO group. This is a completely different protein. And so either a person has Rh or they don't have. So we call them Rh positive or Rh negative individuals. And anti-Rh antibodies are not normally present in the plasma, not like the ABO group. So we are born with antigen and antibodies for ABO blood group, but for Rh, we are not born with it. So a person, an individual with Rh negative blood is going to produce the antibodies only after receiving or coming in contact with a positive blood. If an Rh negative individual never gets it, they will never produce antibodies. So for example, if an Rh negative woman has a Rh positive fetus or a transfusion of Rh positive blood, then the first transfusion or first pregnancy, you don't have problem because it takes almost about 8 to 9 months for the blood, okay, to recognize the protein and produce the antibodies. Okay, so the first child or the first transfusion is safe, right? The first baby is born. But if the same mother has a second child who's also Rh positive, then the antibodies which were produced in the first time, they form memory cells. They will very quickly produce the antibodies against the second child, and it will lead to the death of the fetus, right? So they can cross placenta and they can kill the baby. Prevention is done. Now, of course, we have something called as ROGAM, which is as soon as the mother is tested negative and has a Rh positive baby, they're given anti-antibodies to kill the antibodies that the mother is producing. So that prevents any number of pregnancies being miscarried. So it does bind to the fetal agglutinogens so that the mother will not form antibodies if they're exposed to the child's blood. So we do have that, but it is very, very cautiously. It's considered very, very serious, right? So as soon as the mother gets pregnant, they start the ROGAM immediately, whether it's Rh positive baby or Rh negative baby. So that is your blood typing, and here it is. So Rh negative mother with the first child will produce some antibodies, but the baby will survive. So here you have the antibodies, and then the second fetus will be attacked by the antibodies, and this child will not survive. Okay, all right. So I'm going to stop here and then start the next part, which is the leukocytes.